Hello, everybody. This is Pete Meyer from SP Grid. I'm standing in for Jason Key, and I believe we are now ready to get going. This month's webinar is let's see, Gunnar Schröder, who will pronounce his name better than I will, and he's going to be talking to us about DireX and cross-validated real space refinement density maps. So with that, I will let you take it away.
I will be talking about DireX, basically the program that's developed in my group for the last few years. It does real space refinement, and it was initially meant to work for low resolution map, and kind of the technology was a bit faster than I thought. So now, of course, many people are producing higher resolution maps, and for maps at the resolution that reaches crystallographic quality, you could probably use in a similar way also the standard crystallographic tools like CNS, Phoenix, and so on for the high resolution refinement. So, but I will be focusing more on the cases where you have less than atomic or more intermediate or low resolution density maps, like resolutions much worse than 4 angstrom, maybe 5 to 20 angstrom, something like that. Yeah. So that's the initial target resolution for DireX. But I also show in the end a little bit about some alternative applications of DireX, because it's quite general. It doesn't really need atomic models, or protein structures can work with any type of models, and then that's what I show in the end. I will talk about some of the features of DireX. First of all, DireX is meant to be fast. Yeah, it was initially developed for very large systems, and I based the whole thing on the Kinked algorithm. I will not talk too much about this. The Kinked algorithm is an efficient conformational sampling algorithm that was initially developed to generate quickly an ensemble of structures around a given crystal structure. And I'm using that idea basically to generate new structures based on a list of distance restraints. Okay, and DireX optimizes the correlation between a density map computed from the model and a certain target density map. Is typically your cryo-EM reconstruction, for example. And we have cross-validation in there. We have DN restraints in there, and some kind of other restraints that I will also mention during this presentation. I'd like to mention that this has nothing to do with normal modes. Yeah, some people, if they hear elastic network or so, then they immediately think about normal modes. But DireX has nothing to do with normal modes. It never computes normal modes, and it's not restricted to deformations with normal modes. Yeah, it can typically uses all atoms, although it can also do coarse graining if you want. So the typical situation where we use DireX is shown here. So you have let's say you have a starting structure, a known crystal structure or an homology model in a different conformation, and you have some low resolution data. This here is an 8 angstrom density map, for example. Yeah. So if you just use a standard refinement approach without any additional restraints or so, then you typically mess up your structure if you just fit the structure into the density. And at the time when we started developing DireX, we thought we wanted to use all that information that we already have in the original crystal structure or in the homology model. Yeah? The fact that there is a helix, we want to, most of the time you want to preserve that helix. Yeah? If the data doesn't tell you otherwise, you want to preserve all the details that are in the model. So the idea is, what we did then is we developed this deformable elastic network model, and the motivation was that we wanted to refine only those degrees of freedom that need to be refined to fit the data, but not more. So in other words, another way of saying this is we want to change only the relevant degrees of freedom for which the data actually provide information. Yeah, I will explain how the deformable elastic network looks like. So let's look in the upper right figure where you see the helix and the density map. So in the left part, you see there's the helix doesn't fit the density. So initially what we do is we define a large number of distance restraints in our starting model. That is done by choosing randomly two atoms or atom pairs that are within a certain distance range and connect them with a spring. And then when we start to refine the structure into the density, you will see that some of the springs need to be compressed more than others. So some of the atom pairs need to change their distance more than others. Like the helices, for example, the helix pieces, they fit quite easily into the density without any further deformations. But then there's this one distance restraint, or this one atom pair that I boxed out in this red box. That distance needs to become much smaller. Yeah, in the figure below, where you see the target function and the parabola that shows the harmonic restraints, I plot the energy versus the distance of that particular restraint. And the rightmost restraint, the dashed line, is where the distance is in the start model. And now we allow the minimum of these restraint potentials to move. So we don't just allow the model to deform, but we allow the restraints to deform. So the minimum of this potential can move. And it typically, so we set this up to move, kind of relaxes towards the current structure. So if the structure is moving, then the restraints can slowly adapt and follow the motion of the structure. And the minimum itself is then attached by another spring to the starting model, or can be also another reference model. So some reference model. And you see that there's a gamma parameter that controls the strength of the spring. So the stronger the spring, this gray spring here, the closer you will stay to your reference model, and the weaker this spring is, the more you can fit towards the data. So the better the model can fit to the data. And the gamma parameter is a value between 0 and 1. So 0 means you stay exactly at your reference model with your restraints, and 1 means you completely forget about your reference model and you can adapt to whatever structure you want. Yeah. And there's typically, at low resolution, there's typically some optimum in between that balances the reference model information and the experimental data. And then on the left side, you see here the target function is basically the data and the chemical information plus your DN potential with a certain weight of that potential. So the DN restraints are also implemented in CNS and also in Phoenix. If you want to use refinement with X-ray diffraction data in reciprocal space, for example, this is also possible there. So there are basically three main forces that act on atoms in DireX. One are these Concord restraints, and those are basically only maintaining the correct stereochemistry. So that means that restrains bond lengths and angles and keeps planar groups planar and prevent atom overlaps. That's all these Concord restraints are doing. And the restraints are actually formulated as allowed intervals. So every restraint is a distance restraint in some, except for the planar restraints, okay? But all the restraints are allowed intervals. So because you have a certain interval on all the restraints, that means you have a certain region in conformational space that is allowed and accessible, and Concord samples this allowed conformational space. And there's one point that makes it a bit different from the original Concord algorithm. DireX learns the parameters from the input structure. So you read a structure into DireX, and DireX will learn all the bond length and angles from that input structure. So that has advantages and, of course, disadvantages. The advantage is that DireX can read any molecule you like. So if your protein contains some strange ligand, DireX will not complain. It can read all of that. Other than like typical MD programs or refinement programs, they need some parameterization of all the atoms. That can be complicated if you have weird atoms or weird molecules in your system. So DireX doesn't have a problem with that. But on the other hand, that also means that if you have a bad starting structure that is not minimized and all your bond lengths are corrupted, then DireX will not correct those. Yeah? So you should start with some. You should not expect that you get a better geometry from your model by using DireX. It is not meant for this. Okay, and the second type of forces then is DN restraints that I just controlled, that I just explained, that controls the deviation from the reference model. And the third forces are then the density restraints that move the model into the density that optimizes the overlap of the model and map. So I just showed that example briefly. So this is an initial RMSD here of 13 angstrom. This is a one conformation, and we have a map of that protein here in another conformation, and then run DireX. It fits very well. This is not really a miracle here because this is a simulated density map without any noise. So it just shows you what the type of motions that can be described with DireX. So it has a quite large radius of convergence and can also be used directly to fit such kind of structures. An important issue is at low resolution always is overfitting, yeah, because you have many atom coordinates, you have many parameters, and at low resolution you only have a limited amount of information. So that means you can easily overfit your data. And the typical way of handling this, or at least detecting whether you're overfitting data, is a cross-validation, and that is standard in X-ray crystallographic refinement. The idea is you split your data set into two sets, one working set and a test set. And the test set doesn't need to be large; it only needs to be as large as to give some significant validation value. So in crystallographic refinement, you typically look at the R value. This is defined here, and then it gives it the deviation of your measured amplitudes from the calculated amplitudes from your model. And in crystallographic refinement, you typically choose randomly structure factors from all the different structure factors and define them as your test set. And then you calculate the R value only for this test set that you didn't use in the refinement. So you only use the 95% of your work set for the refinement, and then look at how well your model fits to the 5% of structure factors that you didn't use. Now the problem is with EM densities, it doesn't really work that well, because the structure factors or the Fourier components in your density map are quite correlated. So the neighboring Fourier components are correlated. If you just take randomly structure factors from your whole set, then they wouldn't be independent at all, because the neighboring Fourier components are in the work set and they are heavily correlated. So what we instead do is we exclude an entire range or shell, a Fourier shell from the refinement, and we use a high resolution shell for two reasons. One, it's most sensitive, of course, to changes in your model or errors in your model, and plus it already contains a quite a low signal to noise ratio. So the noise already is quite large in the very high resolution shells. And in practice, you can typically use a Fourier shell that you wouldn't even include in your refinement or in the map calculation, because it contains so much noise. It's a range that that you would not look. I mean, you would basically filter away normally if you look at the density map, for example, because it's so noisy. But we only need it for the validation part. So the actual correlation value that we get from this shell can be very low as long as it is still significant, and as long as you can, during the refinement, you still see an improvement in that correlation. It can be 0.1, something like that, and as long as you see that it's improving and you can still optimize your parameters, then this is still okay. Okay, so a few more words about the technical things about DireX. So DireX is in practice a command line program. I always use it on the command line. We have a Chimera plugin. It's kind of a GUI. We haven't tested this much, so if anyone is interested, then please try to use it. I don't have much experience with that. I mean, and I only explain here how to use it on the command line. So you look at the DireX call DireX and then dash PDB input PDB file. So this reads in the input PDB file, and then with map you read in the density map, and then you define some output. And refine.par is a parameter file, and that contains all the settings that DireX needs to run. DireX comes with a few more tools. That the number of these are constantly growing. I will talk only about one or two of them later on. And so the parameter file is an kind of free format file. It contains some explanations of the individual parameters. It's not that difficult to understand what the parameters are about, and you can generate such a list of all the default parameters and default values with the -OF option. This is typically a good start if you want to start a new project. I have tutorials that you can also download from the place where you download DireX. Those tutorials are different difficulty, I would say. You start with a very simple Kinked Helix at low resolution, and I will look at this in a second. This runs very quickly, and it gives you very quickly an idea of how DireX works. And then there are some other examples that I briefly mention later on. So this is a typical way to run DireX. So again, you run read the parameter file, the PDB file, and the map file. And what I always also do is you can write out, this is the -OX option, and you can always write a trajectory file. So that means it writes every step of the fitting trajectory into a file, and then you can later on look at the dynamics, or not the dynamics really, but how the model moves, during the fitting procedure. And this is typically really helpful if you want to find what's going on and what's wrong maybe with the settings, and this gives you a much better feeling for what's really happening during the refinement. So those are the main parameters that you need to set. So there are some general parameters that just the n steps is just a number of steps, and then we have the perturbation factor, because DireX makes some initial perturbations to the coordinates that's kind of driving the sampling. That's the diffusive part, and that also helps to overcome some barriers. This is the perturbation factor, and the annealing kind of scales down this perturbation factor from 1 to or from this given value here to 0 during the step of, in this case, during 100 steps. And then there are the DN parameters that define these DN restraints. First of all, you set it to yes if you want to use them, and then the number ratio is the number of restraints per atom that are used, and then the restraints are generated automatically and randomly. Then the strength of these restraints, and upper and lower is the distance interval from which these restraints are chosen. So the atom pairs that are within 3 to 15 angstroms can be selected for the restraints. Then the gamma value that we talked about. And you can also make the DEN restraints not be completely harmonic, but become linear after a certain distance of 0.1. In this case, this is an angstrom. And you can also completely break the restraints with the DEN break distance. If you set this to a certain value, then you can make the restraint break. If the change is too large, then you can make the whole thing break. This allows for larger conformational changes. And then you have the density map parameters. One is the map strength. That's just the scales the forces that drives the model into the density map. And then you have to set the resolution. And you have to set the Kerr map d min and Kerr map d max is the resolution range that's used for refinement. So the higher the resolution, in this case, the 10 angstrom value, that really defines the cutoff of the resolution that is used for the refinement. And then the map CVD min and map CVD max, that defines the resolution range that's used for validation. That's kind of our test set or the free set. Typically, we use, in this case here, this is set, the higher resolution threshold is 8 angstrom, and the lower one is set to -1. That means it's the same as Kerr map d min. So in this case, the free set is 8 to 10 angstrom. I think I have it here. Oh yeah, this shows the values. So in this case, the work set here is, or the green range that's defined by the Kerr map values, and the red range is defined by the map CV values. And as I said, you don't really need to include that much of information for the validation. The correlation can be the C3 value. This is the map correlation that is computed only with this free Fourier shell. And that can be a small number as long as you still see significant differences between different models. Yeah, the only reason you want to use this is because you want to distinguish between different models and want to decide in the end which model is more correct than others. And as long as the C3 value still gives you a significant difference, then this is enough. We talked about this already. So you can look at the fitting trajectories here. I typically use VMD to look at the fitting trajectories because it's a much faster has a much faster way of displaying the trajectory. And the reason directly this XTC format is a compressed format. And yeah, and then of course you look at the model map and the density map in the end. Just very briefly, those are the values that are in this map CC file that writes out the cross correlation of model and EM map. And also this is the C work value, and then the C3 value is the same cross correlation, but for the free maps. That those are the maps that are filtered to the free Fourier shell, and then some other like real space R values, reciprocal space R values, and masked cross correlation values that you can look at. I also find it usually very helpful to look at the model map because this is what is fitted in the end. Yeah, you optimize the agreement of the model map to the target map, so you can also write out the model map directly. Just quickly, what you would change, for example, if you go to high resolution now, if you the same Kinked Helix example here at high resolution, the only thing that you change basically is the gamma value in this case. I set this here to 1. If the resolution is high enough, you don't really need any reference model anymore. You can just directly completely fit your model into the density. And you can also make your whole model a bit more flexible by lowering the interval from which you choose the restraints, the DN upper and DN lower. And then there's this DN sidechain parameter that scales those restraints that involve sidechains. So you can make your side chains more mobile if you lower this number. So we set this to 0.2 here. That means the forces on the DN restraints are basically scaled by a factor of 5, and they are weaker by a factor of 5 than those in the backbone. And the resolution, of course, then also needs to be adjusted to, in this case, 3.3, and then the free map is set to the free shell is the range from 3 to 3.3 angstrom in this case. I want to make one note on preparing the EM density map. We have a tool that's called Wisdom that sharpens the density map, and it does that by applying a structure factor distribution and a density histogram from a statistical distribution of atoms in a protein. So that also takes into account the shape of the density map. So we basically fill the density map with the statistical distribution of atoms that you would expect from proteins. So you have to define, give also here the mass of your protein. From that, it determines or estimates the volume, and then it determines the structure factor and the density histogram and applies those to the map. And this usually gives you a better balanced density map, and also and this is also part of DireX. This is the tool is called DireX Wisdom. And maybe one note also on the typical problem that happens if you have a large protein complex that consists of individual chains, and you have individual models for these chains, and now you want to combine this into one large model, then oftentimes that doesn't really fit without any clash. And many programs will have some problems with heavy clashes between models. And in Concord, in DireX, you can, there are some options to make sure that the chains do not see each other and they are allowed to overlap. And then you can also add some repulsive forces between the chains while fitting into the density that drives them apart from each other. And once that is accomplished, you can then switch back to the normal refinement and let the interfaces click in, basically, and then let the models refine. This is explained here. So this is in the parameter file. There's an option that tells you to add a repelling or also attractive force if you want to different chains. Another thing that is useful is oftentimes in a larger protein complex, there are some regions of your model that are not resolved that well, and maybe some loops are missing, or some entire domains have very weak density. Then this missing density can lead to an artifact in the fitting of your model. Yeah. So of course the entire model wants to fit into the density somehow, and this is why we implemented this occupancy refinement. Well, this is basically something like a B-factor refinement, but we're not really applying a B-factor or atomic B-factors here. This is a bit simpler way of handling this problem. Is we just scale the contribution of atoms to the model density. So, and this is why I call this occupancy refinement, because it's much closer to what occupancy refinement does. So here are some parameters that are needed for this. So the map refine occ means you're refining the occupancy values, or the scaling of each atom mass, basically. And you can also have restraints. So you have group restraints. That means you can scale atoms within one residue the same. So all the atoms within one residue have the same occupancy values. And in addition to that, you can also have restraints between neighboring amino acids, so that that even lowers the number of parameters even further. But you always have to realize or understand that by this you're adding many more parameters, and it's much easier to overfit your structure by introducing additional B factors or occupancy values that you're refining. So this will typically doesn't help to improve your C free value. Yeah, so you have to be very careful with adding more parameters, because then you overfit much faster. DireX can also read very general distance restraints. So it just takes one file here, and this is shown in the disre.dat, a file with 2 atom IDs, the distance between them, and the strength of the restraint, basically. And with this, you can define any type of distance restraints. And what we often do is, if you want to preserve specifically hydrogen bonds, for example, or enforce even further the hydrogen bonds in your structure, you just write out a list of all hydrogen bonds and put them into a distance restraint file, and then you can add those in addition. And I wrote down here an easy way to write down the H bonds from Chimera, and then you just need a small change in the file format to read this into DireX. And in the parameter file, there are these XD strengths values that control the strengths of these restraints. And in a similar way, you can also define symmetry restraints. I call this NCS restraints because it's similar to the concept in crystallographic refinement. You don't define here a specific symmetry operator, so you cannot really say, well, this domain is, I don't know, 10 angstrom away from that domain and rotated by this far. It only applies, it's basically similarity restraint. So what we do is we keep the domains similar, but they can be anywhere in space. Yeah, so that the relative position and orientations are not controlled here. And oh yeah, I should also mention that because DireX uses these distance intervals for bonds and angles and also distances between atoms, the geometry that you get with DireX in the end is not optimal. I mean, it's usually not very bad, but it can be better. So what we always do is, in the end, we do an additional energy minimization, or run some other refinement on the structure to make sure that your structure is the geometry of your structure is the best possible. I would like to show very briefly example of a typical application. I would say maybe not a typical, but a nice application of where we use DireX. But this is a collaboration with Niels Fischer and Holger Stark in Göttingen. They obtained about 50 different conformational states of the ribosome, and with the tRNAs in different conformations. And so I don't have really the movie. I just show, just a few snapshots here. So where we see that the tRNAs are moving through the ribosome. So we have many of these states, and then we fitted a structure of the ribosome with the tRNAs in DireX. So what we obtain from these lower resolution density maps are a series of atomic models where you see that how the tRNAs are basically moving through the ribosome. I just show a few of these structures. We refined 24 of those in total. And then in collaboration with Helmut Krupmüller in his lab, they performed MD simulations to study the connections or the transitions between these individual states and determine energy barriers and so on. But the nice thing is in DireX is relatively how do you say that? well, DireX doesn't really require a complete model. Yeah, it doesn't need a full atomic model. It can work with basically any type of input. And in this case, here we just use a C-alpha trace. We can add additional distance restraints to impose some secondary structure information and just refine the backbone trace very quickly into density. Yeah, that's a quick and easy way to generate backbone traces or optimize backbone traces, maybe generate a number of backbone traces that you can use to start building full atomic models, for example. But this way, we can also sample different conformations very quickly. And another thing, this is the last point that I want to show you. Because DireX can read so different types of models, we thought we just treat the entire density map as a model that we refine in DireX. And so the goal here is an elastic alignment of two density maps. So we basically put beads or point masses at each voxel in our density map. And the masses are chosen to be proportional to the density values. And then all the beads are connected by harmonic distance restraints, which means we have an elastic body, basically. I mean, the whole idea is that we want to use, in the end, this elastic alignment to average density maps in different conformations to improve the resolution, of course. Yeah? If you have 10 density maps in slightly different conformations, with the elastic alignment, you might be able to average all those maps and align all the individual domains much better than you could do with rigid body alignment. So I'm just showing this here for a test case. This is a domain motion of this is a small protein. This shows here the density grid itself. Yeah, this is cut out in the region where we have actual protein. The color is the density value at each density map. And now this model here is put into DireX, and the target density map is not shown. So the target density map is in a different conformation, and now we are deforming the grid itself. Yeah. See that locally the grids are preserved, but globally the whole thing changes. And this is a pretty drastic change. I think in practice, if you want to apply this to real data, you probably wouldn't use such large motions, but it's just to show you the principle of this. So this shows here the grid deformation itself. Now we can, for each step during this fitting trajectory, we can now compute a density map from this grid. Yeah, and this is shown here. So this is the same trajectory, the same motion, but now we are looking at the density map, how that deforms. That's based on the deformed grid. Yeah, now locally the density map doesn't really change that much, but it's a global deformation that we are interested in here. Yeah, so that was it. You can download DireX and the tutorial files here from the SimTK website. We have documentation also on our lab website. And I also want to advertise the upcoming workshop that Klaus Schulte and I are organizing in Jülich this August. So we'll have DireX tutorials, we have MDFF tutorials, and Frank De Maio will also present the Rosetta tools for cryo-EM modeling. And it's a CCAM workshop, so you have to go to the CCAM website here if you want to participate. Okay, then I'll thank you and take questions if you have any.
I do have one question, though, and I apologize if this is something that you mentioned. But is it meaningful to compare C-free versus C over? Does that give you anything useful?
Yeah, not at the moment. The problem is that you're calculating this from different Fourier shells, and they are not comparable. Yeah, so typically the C-free value is always much lower than the C-work value. But we are in the process of defining kind of a normalized C-free value because we can determine what is the practically the lowest C-free value that you can expect due to correlations in the data, and also we can also estimate what is the highest C-free value that you could get based on the signal to noise ratio in the shell. And from this we can determine a normalized C3 value that gives you and then an absolute measure of, yeah, kind of a more absolute measure. And I think this this will be very useful, but this is not at the moment implemented, and it's also we are still working on that. So this will be hopefully the way of doing this in the future.
So I have a question. If you don't have a homology model, can you just fit in as a polyadenine chain to the density?
Yes, yeah, that's definitely possible. Yeah, yeah, it doesn't need to be the correct structure or the yeah. So we do this as well. I mean, this is the first step of building the backbone. We first trace the model, and then we build all atom models, and we refine them with DireX into the density and get good placements for the C beta atoms. And then we can think about the sequence assignment, for example. Yeah, that's possible.
I just have a quick question. Just typically, what's the good numbers for the C and the C3?
A good number for the C3, you mean?
Yeah, yeah. So how do I know from the C numbers that the refinement is good or not?
Yeah, as I said, so it's very hard to put it at the moment to put an absolute value on this. So what we typically do is we optimize our restraints to maximize the C3 value. Yeah? So with this, we get optimal restraints and make sure that we are not overfitting. But the absolute value of the C3 is not relevant, really. Yeah? It's just of relative importance. So we only maximize that value, and then we know that we have the optimal restraints.
I don't think we have any more questions locally, and I don't see any in group chat. So Gunnar, thank you again. And everyone, thank you for sticking around until we could get started.